It's always difficult.
Okay, let's get started, everybody. I think, hello, nice to see you all again. So today, it's my pleasure to introduce Tobias Kürschner. Tobias did his PhD at the Leibniz Institute in Berlin, and studying the effects of global change, dynamic resource landscapes, and individual movement, and how that affects the spread and evolution of classical swine fever on wild boars. And so now he's a postdoctoral researcher at the Bavarian Forest National Park, where he's working on pathogen spread in vertebrates and things like liver flukes and things on African swine flu in wild boars. Over to you, Tobias. Welcome.
Thank you very much for the nice introduction, and thank you for having me here. So, from an ecology perspective, pathogens have always been a challenge. So from, of course, pathogens that probably most of us know that strongly affect or affected human populations, there's also the side that they usually have a zoonotic origin. So there's some animals are involved. And of course, while we have pathogens that directly affect humans, we also have a whole host of pathogens that might not have a direct effect on humans, but strongly affect individual wildlife populations. So just to name a few, I mean, the African swine fever has been quite prominent in the news over the past, just purely due to its devastating effects. And one important point when looking at diseases is that the resolution or the scale at which we look at the system matters a lot. So if we imagine a population just as on a very broad scale, just individual points in a arbitrary landscape, we don't really see all that much. And here it is important that once we increase the resolution of the system further and further, we at some point can see a complex network of interactions between individuals, and those interactions will shape how pathogens can be transmitted through the population or to landscapes. And the problem here is that in most cases the transmission of pathogens has been modeled in a density or frequency dependent way, which arguably sacrifices realism, especially when assumptions about how individuals come into contact are not taken into consideration. One of the classical approaches here is just assuming homogeneous mixing of populations, which just means every individual of a population is in contact with every other at every time. So obviously that in nature is not the case, but what we have in both humans and in livestock and wildlife is so-called non-random association. So there's contacts between specific individuals, specific groups, over specific distances. And those contact networks are a key point when it comes to trying to understand how a disease spreads through a landscape. So those contact networks are very important. However, they also pose quite a big problem. Because while they on the one hand represent all those pathways I just mentioned, they're also extremely difficult to gain knowledge about. So it is very difficult to go into the field and have perfect knowledge about which individuals of a population come into contact when and where. Especially when external drivers such as global change also affect that system. Can be through climate change or just land use change, habitat destruction. There's a variety of factors that can change the density and distribution, and to some extent also the susceptibility of individuals to pathogens. And in return, this will could increase the frequency of outbreaks, but also poses the risk of an increase of spillover events between wildlife and, for example, livestock or humans when we, for example, habitat gets reduced and humans or farms get built closer to wildlife populations, there is a higher risk of spillover effects. So one key point here is that animals, of course, have also mechanisms to deal with changing landscape by themselves. And here, movement of individuals is a prime adaptive trait, how they can react to changing circumstances. However, it is also important here to keep in mind that not every movement is the same. Not every individual of a species moves in the same way for the same reasons. And here behavior of individuals matters a lot in shaping movement patterns and therefore also contacts. So individual decision making is important. As I just said, not every movement is the same. There's always a decision making process within the animals, where to go, why to go there, and when to go there. So in terms of disease dynamics for us, that is very important information to know about individuals, because that can give us big indications on how diseases spread through a landscape, and more importantly, how those individual decisions can form complex system-level patterns that we then can analyze. And one important tool that we can use here is modeling, because one big problem we have when it comes to researching wildlife diseases is that there is very little experimental work we can do that works on a large system-wide scale where we can see spread patterns, for example. So we use individual-based models to simulate how individuals behave in landscapes, and in return how that affects disease spread, for example. So what is an agent-based model? So quite simple. Instead of looking at an entire population, what we are doing is we are looking at every single individual of a population, which has its own set of traits, of variables. We then, in return, get these emergent population scale patterns. Just a little example here is for here. in the bottom, there's isolated habitat patches in the landscape, and here all individuals' movement is simulated, and what we can see as the resulting pattern is how are those isolated habitat patches connected, where are those avenues that most individuals travel, and that can of course be scaled up to an entire habitat. So what are some of the more concrete things we can do? Approach is just plainly looking at how a disease is transmitted in a landscape and how it persists in a landscape with wildlife populations. So here we were looking at resources. So imagine the landscape as having a certain amount of resources, and ideally a species will have evolved its population dynamics to follow the resources of the landscape. So when the resources are high, their need of resources is high. But what we already can see in some wildlife populations is that there is a shift where those two curves move away from each other. So migrating birds arriving at their overwintering sites before the local food source is at full abundance. And this mismatch can, of course, affect the population through first survival, but also it can result in a change in movement patterns. So because the area they're usually used to for high resources, suddenly when they arrive has no resources. So they need to find a different resource patch. And this in return will affect how a population is distributed in a landscape. It will also change, increase or decrease local densities at specific points. And in return, this will, of course, affect how a disease will be transmitted throughout the landscape. So if suddenly a lot of individuals are clustered in one small area, this is a very good part for disease to be spread between a large amount of the population. And then we can use that information to see how long would the pathogen be in a population, for example. There's a lot of other things we can look at here, but just to look at one example here is how can this movement reaction to the changing resources affect the population, and can it reverse the effects that this asynchronous resources have on the population? So for this system, I was looking at the European wild boar, which is probably known to most people as a group living social animal, and is basically distributed all across Europe. And on the other side, we have the classical swine fever virus, which is an important, is a directly transmitted disease, so from individual to individual. And it has both acute and chronic forms, which can stay for quite a long time in certain populations. So we were looking at this specific shift in the resource availability in relation to the host population. So here, when the host population has, the host reproduction is at its highest, so when the most juveniles are introduced into the population, the available resources are not at the maximum. So usually those would then affect the population through either the changed movement, but also through mortality, obviously. So if we then put this into a more theoretical landscape, here's just two theoretical landscapes. They are clustered, and the grayscale is kind of the resource availability, and each cell somewhat represents a group of wild boar in the landscape. And we're looking at, on the left here, the ideal scenario, the resources and the population are perfectly in sync. And on the other side, the extreme case, where there's a complete mismatch between resources and population. And what we expected to see is that the population cannot persist when the resources are completely asynchronous to population, because the increased mortality due to the lack of resources, will cause the disease to basically burn itself out because it cannot find enough suitable hosts to stay for a long time in the population. However, what we saw is here now in a color, we can see how long the disease was present in a group of wild boar. So instead of the expected dying out of the disease, at the super asynchronous level, we saw a spatial clustering of disease hotspots in certain areas of the landscape, where the disease could persist long enough for the resources to swing back up and allow new naive hosts to be introduced into the landscape. And then it was able to survive for over 50 years in the landscape just by retreating into those hotspots at time of low resources. And from that, move a bit to more bit of a practical application. While directed transmitted disease are relatively easy to simulate, there's also more complex forms of diseases, or in this case pathogens. So another thing I want to talk about is invasive parasites. So here is the great American liver fluke, which is a relative of the normal liver fluke. It's an invasive species that is very prevalent in the eastern parts of Europe, and it has at this point also reached large parts of southern Germany. And the difficulty here is that, as most parasites, it has a relatively complex life cycle. And it is a parasite in most cervids, where the red deer in Central Europe is their definitive host. The red deer will excrete the eggs. They then need water to hatch. In the water, they will evolve into motile larvae, and they will actively seek out, in Central Europe, it's 2 specific types of aquatic snails. In those aquatic snails, they will then multiply themselves, and usually at night being released by the snail, where they will then go onto aquatic plants and insist themselves on them, waiting then for cervids to graze upon those aquatic plants, and thus the cycle continues. So in red deer, it has relatively little effect, especially in the early life stages. There's some theories that it might have some more adverse effect when they accumulate over time in the liver of the red deer, but there's a very minute effect on them. Similarly, for wild boar, there's a bit stronger effect where there's weight loss and lethargy. However, and importantly, it has a very strong effect on, for example, the roe deer, where the liver fluke cannot properly migrate and encapsulate themselves in the liver, and that is usually, or almost always, fatal for the roe deer. And this is a problem because roe deer is one of the prime prey species for the European lynx. So roe deer population that is being devastated by an invasive parasite can have then cascading effects further on into other species. And for this, we're going to the Bavarian Forest National Park, which is located in the southern eastern part of Germany at the Czech border. And a few maybe important things to note is that there is a so-called management zone here in the purple areas where there is population control through hunting, mostly to prevent large amounts of red deer, for example, to exit the park and then destroy surrounding farmlands. To keep individuals mostly in the park, especially over the winter where there is low amounts of resources, there's also 4 enclosures which are opened. The red deer are fed to go in there, and then they're closed over winter and keep them in that area, and then in summer they are released. However, that also gives us a few very dense contact points here. While there is not very much transmission of the parasite throughout the winter, those enclosures are also kept open in summer, and many red deer have gotten used to that. So it's a nice, safe space. They can enter and exit if they want to, but in summer, all the accumulated feces in the area will, of course, be a concern in terms of parasite infection. And so we were interested to see if we restrict the access to those enclosures, especially during the summer times when the infection peaks, could we, through a relatively simple measure, just closing a gate basically, over the long term, prevent or reduce infection rates. So we also modeled all the red deer in the park, and just very quickly, so over a long time period here, we can see the infection cases recorded only through hunting. So we employed the same methods as we would also do in the field. So we only know if a red deer is infected when it was hunted, and then the liver is dissected to see if there's liver flukes present. So we virtually, so to speak, also employ the same sampling scheme as the hunters in the park would do, by only removing areas out of the management zone and also in the same quantities as the hunting in the past has been done, to really see if we could recreate those patterns. And in blue here is what happens when the enclosures just stay open. So they can freely enter or stay in there the entire summer. And in red, we see what happens if we close them in summer. And after, well, some time, over the long term, we can see a reduction or a decrease in the infections of red deer. So that could actually be one of the possible management solutions that is relatively easy to implement and doesn't involve large-scale population reductions, because density, of course, is also a factor. If there's no red deer in the park, there's no liver fluke transmission in the park. But this, of course, is not an option. So finding those solutions can help. Which brings me also to the next part, where we then, on a different topic now, are trying to integrate management practices more detailed in wildlife disease models. So this is a project is currently ongoing, also in the national park. And here we were looking at the African swine fever. I mentioned it briefly earlier. So it's a highly virulent disease, and it's almost 100% fatality rates across the board. And of course, wild boar are the ones transmitting it, some degree. And it can have substantial impact also on commercial swine industry, for example, when it reaches farmland. And integrating management on a very small scale into disease models is always challenging. So on one hand, we need to incorporate the individual responses of individuals to management interventions. Suddenly, if you build a fence through the home range of a wild boar, there's bound to be some reaction by the individuals to that. Or if you start to hunt in a large scale, there's also expected to be some reaction from the wild boar side. On the other hand, you also have to create robust enough algorithms that represent the group's movement or the individual's movement good enough in response to external factors that you are not basically making up the movement as you go. So ultimately, what is of interest here is the African swine fever dynamic in the population and how it spreads spatially. So for this, we use a a very large amount of databases, of data, telemetry data from wild boars across Europe. There's currently several experiments ongoing, which I will mention in a bit, to basically inform our model. We then combine that with spatial data, such as currently developing a new habitat suitability map for the wild boar for the entirety of Europe, which will then also be used to inform detailed movement decisions. And those movement decisions and decision-making in general will then be also put into context of varying management solutions we can apply to the system to then have a reaction from the individuals. So what we are getting here is a very detailed, scalable, and spatially explicit agent-based model for African swine fever management. And I'm not going to go into too much detail. First, it is still under construction, and also it is a relatively complex model. I'm only going to focus on some of the compartments that could be of interest here. So, of course, importantly, wild boar groups wild boars live in groups, so-called sounders, and they're usually relatively spatially fixed to a certain area. So a group will not move out of their home range much. So we use telemetry data to identify certain patterns in the movement of wild boar. So what we could find in that data is that there's dynamically changing points of interest where those wild boar groups within their home range tend to go to, accumulate for some time, stay there for some time, and then they move on to a different point. This can be feeding sites. There's a varying sites here that they can visit. And they also do excursions outside of their home range, but they always, in most cases, return. This is just purely for the groups. So we were able to identify how many of those points a group usually has over a certain period of time. And then we can simulate the group's movement within each home range, and we get a variety of different space use patterns that are represent relatively well what we also see in the telemetry data. This is accumulated over a long period of time, so it looks a bit chaotic, but if we break it down into the actual parts where we usually have tracking data for, we can see those points quite clearly. And another important part with the wild boar is that the males, once they become young adults, they usually disperse and move relatively freely throughout the landscape. And then especially during rutting season, they tend to visit several closely, spatially close groups. So we also use telemetry data here to inform the movement of the male individuals, because we also saw, or we can also see, that they tend to avoid each other. So while they might be visiting the same groups, they will do their best to not run into each other. So we were able to also recreate these movement patterns in our model. And from there on, we also decided to take a different approach when it comes to hunting, because pretty much every wild boar population in Europe is hunted. And usually when hunting is modeled, it is a flat just reduction of X individuals over a certain time period. What we wanted to include here is the spatial aspect of hunting, especially when it comes to hunting as a possible management solution for stemming an ASF outbreak. Because it is quite important where the individual is in relation to possible hunting spots. So we used in our test cases the actual positions of high seeds in the landscape with an average range in which there could be hunting from them. combination with very detailed hunting data we have for some areas to get the numbers of hunted individuals correct. And here, it is important, we are still trying to get some data from hunters, which is not that easy, to inform something like hunting effort. So how many times does the hunter have to go to a high seat to actually hunt a wild boar? So it's all data that we don't really have a good basis for. On top of that, very important, how does a wild boar react when it's being hunted or when there's a shot nearby? So we did some disturbance experiments in the past, just with the presence of humans, which didn't yield any exciting results in terms of something we can actually use. So actually, right now, there's new experiments going on where people are going to go close to a collared wild boar and shoot in the air, and then we can measure flight distances and flight time to then also use that in our model. And that is, at this point, still ongoing. And for ASF, one very important point is that the direct transmission between individuals is assumed not to be the driving force of infection. One big part here are carcasses. So carcasses of diseased wild boar stay infectious for a long time, pretty much their entire time of decomposition. And so obviously we have to include that also in our model as a possible point of interest. And there's parallel also a study currently being conducted about how likely is it actually for a wild boar to find a carcass? And if they come across another wild boar carcass, what is their behavior? What do they do? And little spoiler alert here, not much. They don't really care, really. We could not observe much contact, so there's not really any feeding on the carcass. They sometimes look at it, sometimes they don't even get close. They just ignore it most of the time. So we are also currently looking into that in terms of mode of transmission of ASF. And that brings me also direct to the modes of transition we are using in the model. So we're also having a temporal dynamic ASF transmission. So while I just said that the transmission directly is not very strong or very common, especially in the early symptomatic stages of the infection. So there's very little in terms of shedding of the disease or the virus going on early on. But then in the late stages, close to death, there's a very high viral load. So we also have this dynamic put into the model. And then, of course, we do have, because there is some observed data, even though it's very little, of direct contact to a carcass. If that carcass would be infectious, then there's a pretty much a guaranteed transmission. And then we can take all this, and the plan is then to apply various management strategies, hypothetical vaccination, or spatial restriction, and just density reduction through hunting in specific areas. But what we can do already is we can look at what kind of patterns on a larger scale does do those interactions and do those individuals form in the landscape. So I just picked out like 3 groups and some individuals here. So we have 3 home ranges of wild boar groups here. They occasionally, not temporally, but spatially overlap. So if there would be an infection going on, there's a chance that an infected carcass from this group ends up in the territory of another group as one possible mode of transmission. And on top we have then roaming individuals, either males or females dispersing, that connect this to the wider landscape. So there's, of course, way more groups all around those I just show here. Just is going to get a bit too messy if I show the entire landscape. And if we then look at where and how often and how strong do we see interactions that can be a mode of transmission, we can get information on detailed contact hotspots, contact networks, and where in the landscape is it likely for disease transmission to happen, for individuals to meet, and where to focus possible management approaches. And this is also something that is largely overlooked in many epidemiological models that don't take those detailed individual contacts into account. So, in conclusion, it is very important to account for those interaction networks and especially individual behavior when it comes to understanding where and how to focus, for example, management, especially since those associations are in many cases not random. They are between specific groups, between specific individuals, or specific parts in the landscape. There could be a feeding site that attracts multiple groups of individuals. So it's not a random point where they meet. And since we cannot really do much in terms of large-scale disease analysis in the field, individual models or individual-based models can be a good tool here and are very important for targeted management specifically. And with that, I would like to thank a lot of my collaborators and co-authors, and I would like to thank all of you for listening to me, and I'm open for questions.
Thank you for that. That was awesome. Do we have questions?
Yeah, thank you for your presentation, very interesting. Yeah, I have several questions, but first of all, I wanted to know, because at the beginning you were showing how the synchrony between the resources and the abundance of the animals, and I was not getting well. So when you have more resources, you have you were showing that there was like a synchrony, right, between
Yes.
the animals and the resources, and I didn't get well why this was. So the relationship with the infection.
Yes, so we assume that, for example, in this case for the wild boar, they have their breeding or their breeding peak, their reproductive peak at a specific time of the year. And usually, shortly after that, their needed resources are also high. So what we're assuming is that it's kind of evolved together to be the breeding peak at that time because the resources to wean the young are high at that point. So that's the relationship between those two.
But then do you get a higher rate of infection when you have more resources?
It's not per se a higher rate of infection, it's just the spatial pattern of the infection changes. So it becomes more clustered in the higher dense areas, if that makes sense.
Yeah, so but because I was wondering, when the animals have more resources, they are like in better health conditions, right?
Yes, of course, that is an important part. It's not included in this specific model because also it is very difficult to gain good data to use for modeling in terms of immune system, immune response to a disease. When, of course, they are on starving, they will have a stronger disease course, for example. But that is very, very difficult to include in a model without the appropriate data, and especially on stuff like the classical swine fever, which this was about, as far as I know, doesn't really exist in terms of how strong would that effect be. Okay. But it's a very good point, yes.
Okay, thank you.
It's behind you.
Hi, thank you very much also for me for this lovely talk. It was really interesting. And I sort of have a related question to your answer you just gave to Irene, and basically about the parameterization of the models. Is that, yeah, especially the last model you showed, that was super complicated. I mean, it had all the, and so what is the trade-off between having a very detailed model in this case and, like, you know, having uncertain parameters, and how do you incorporate uncertainty? And when, you know, when do you say, okay, I try to model it, but with high uncertainty, or I just don't model the process at all. How do you decide these types of things?
That is always a very, very difficult deliberation to see what parameters we can actually include properly. So with the last model, we're actually trying to gather the data to inform the important, or the most, what we decided to be the most important parameters. But what we do a lot is we are looking at outbreaks on a smaller scale to see if we can derive parameters from real life outbreaks to then apply to a modeling scope. Like in the first part of the classical swine fever, there's some good outbreak data. So like the speed, how fast it spreads spatially. And if we can match those patterns quite well with a certain set of parameters, we can then decide if that's enough or not. However, there's of course a lot of confounding factors that we just cannot control for. So there will always be some uncertainty in the parameters that we use.
Yeah, thank you. Do you validate the data then?
Yes.
With the actual observed
in the case of the last model, for example, with telemetry data from wild boars from all across Europe.
Yeah, makes sense.
To figure out those patterns, at least.
Do you compare the results that you obtain from your very detailed individual-based models with the results that you can get by considering a random movement pattern, for example, or a mean field approach, so that you can quantify how much adding that level of complexity increases your predictions?
Yes, we actually did that with the models. I didn't show it here, but we always use complete random movement as to validate, to see our movement, and also not technically mean field approaches, but we have some sort of neighborhood infection, direct spread, more akin to homogeneous mixing at the borders of cells, to compare that to our transmission through movement processes. So that's our,
I actually had a question about the life cycle of the fluke, because it seems like it has like three dependent species, right? And I was just wondering, in pathogen examples that have such a high number of kind of dependent species, how do you consider? So, because the focus is on the mammalian host component, how much information is there or work on other host systems that a pathogen might be passing through, and how much interdependency is there?
That is also relatively difficult. So we are focusing here on the mammalian side, on the red deer side, because that's the mode of transportation mostly. So the snails don't travel distances, so it's the red deer that transports it through the landscape, famously. And so the problem here is that there is some other cervid species that where the flukes also can reach maturity. So there's of course some uncertainty in terms of other species that might transport the parasite as well. So it's not, of course, we cannot capture everything. Would be nice, but then the complexity kind of gets out of proportion.
Anyone else? Oh.
You mentioned that some of these are recently invasive and some of these are old. How recent are these and do you expect these dynamics to change as they become, I don't know, as they've been here longer?
Well, I mean, ASF is sporadically always being reintroduced, and we're at this point kind of thinking we will not get a full-scale wave going through Central Europe, but more like punctual outbreaks. The liver fluke is, I don't know the exact year, I think it was around the 90s when it first arrived in Germany, at least. In the Czech Republic and Eastern Europe it was earlier, but I don't know the exact dates there. So this is relatively slow spreading because the life cycle of the parasite, but it is also not really containable to a level where this will slowly but surely might make its way, depending on if there is a possibility of management at some point to stop the spread. But apart from really having the carrying individuals stop at some point or stop their movement, it is very likely to spread. And classical swine fever is currently there's no cases as far as I know, at least. But all the measures that are used to prevent African swine fever, like fences across borders, are, well, not really effective in stopping wild boar, but stopping everything else. So it's just a matter of time until the next. I think right now there's an outbreak in Saxony in Germany of ASF, and it is very difficult to contain wild boar spatially, so there's most likely to be more to be seen of.
Anyone else? Okay, then I think if not, we finish a little bit early. Thank you so much, Tobias, again.
Thank you very much.
Join in saying thank you again, and we'll see you next week. Thank you.